Thank to the organizer of this program, this webinar 2020, and Dr. Mega for inviting me to deliver a talk in this program. And though it was a very short notice, but I am happy to deliver the talk today. So today's topic of this lecture is the nanostructures and the nanomaterials and how we can characterize them and what are the basic properties of these nanostructure and nanomaterial. So the content during this next 1.5 hours, we will be discussing about these about the nanoparticles and nanostructure. What is nano? What is nanoscience? Is it natural or man-made? What is nanotechnology, what are the properties of the nanostructures, what are the various types of nanostructure, how to synthesize these different nanostructures, how can we characterize those nanostructures, and what are the applications, various applications of the nanoscience and nanotechnology and nanoscience in our daily routine. So first is what is the nanoscience actually, or the nano. Whenever we talk about the nano, what is exactly the nano? So if you see, we generally see the meter scale. Then there are the millimeters in our small, small scale that we keep in our drawing, routine drawing box. Millimeter up to millimeter, we can draw the things. Then there is, if we divide the meter with the thousand, then we get the millimeter, and again if we divide 1 millimeter by the thousand, then we will get the micrometer size. So most of the bacteria bodies, RBCs, and the various organisms, they lies between the micrometer to millimeter in this size. But again, if we divide this 1 micrometer we again with thousand, then we will get the 1 nanometer in size. So in this nano scale is, you can say, the further division of the micrometer size, and generally 10 raised to power minus 9 meter. 1 nanometer equal to 10 raised to power minus 9 meter. So if you see the antibodies, virus, these are the so small, small things that lies in the nanoscale range. Glucose molecule that we talk about, the glucon D and everything, the glucose is a biomolecule which has the size of about 1 nanometer in size. So, and the interesting thing is that this our electromagnetic spectrum, mostly the UV visible and some infrared, they lies in between the nanometer to micrometer size as well. So light plays a very important role. Size and the light, those are the two important properties that will lie with these materials having the nanoscale size. Next, now because it is the new, totally new things to you, so I will tell you some terminology that we will be using in this presentation. One is the nanoparticle. So any particle or the substance which has the at least one dimension in the 1 to 100 nanometer in size. So the important size of any particle for the to be effective as a nanoparticle, it should lies between the 1 to 100 nanometer. So nanoscience and nanotechnology, that relates to the nanoparticle, and we call a particle nanoparticle if it has at least one dimension between 1 to 100 nanometer. Next term will be the nanomaterial. What is a material? Material is an object. Particles are different, objects are different. So nanomaterial, any material which has this, which also has a size, or you can say the at least one dimension in from 1 to 100 nanometer, then we call it a nanomaterial. Now coming to the nanoscience. What is nanoscience? So nanoscience is the study of the fundamental principles of the nanostructures, obviously. So what are these nanomaterials between bulk to atomic? Here, this size more than 100 micrometer, generally we call them the bulk material. So between the bulk material and there is the, if we go to the angstrom level, then we call it a molecular level. So the nanoscience deals with the nanostructures of the nanomaterial in the size of the 1 to 100 nanometer in size. And nanotechnology, what is nanotechnology? It is the application of the nanostructure to make useful device for us. So nanotechnology it is more related to the engineering science. And if we are able to device if we are able to make the useful devices using the different nanostructure, then the nanotechnology comes into the picture. So there are the four basic things: nanoparticle, nanomaterial, nanoscience, and nanotechnology. So yeah, as I told that the we will be interested only between the 1 nanometer to 100 nanometer in size, where the most of the proteins, DNA, viruses, those have this equal size of the 1 to 100 nanometer. And various types of these nanostructures have been designed: micelles, liposomes, dendrimers, gold nanoparticles, quantum dot, polymer nanoparticles. They are generally, their size is restricted between 1 to 100 nanometers, so we can get the desired property of those nanoparticles. Now, is the nano is the natural or the artificial? Nano, it is not that the nanoscience which was recently has the nanotechnology or nanoscience has started just 3 decades before. No, it is not like that. Nano is already available in the nature. If you look at the lotus leaf, in the lotus leaf, it lies on a dirty surface, but still if you see the lotus leaf, they are always clean. Why they are so clean? Because of the nanoside wax papillae present on the surface of the lotus. So these nano, they have the papillae, wax papillae, which have the nanostructure size, which make them the super hydrophobic. And whatever the things will fall on it, it will immediately form a drop and it will come out. So lotus leaf has the nanostructure size wax papillae. Similarly, there is a Tokyo Gecko. So this if you see this specific type of, it is a type of a lizard which has the cities in the micron size, but later on it was studied that these cities which are present in this particular lizard, they have the nanometer scale projections. So that nanometer scale projections help this lizard to stick to the particular surface. So those projections are also in the nano size. And there is another phenomena that we call the iridescence. Iridescence is a phenomena which you can say that it is related to the structural colors. Some surfaces have the different structures. Those structures are made up of the micrometer size and the nanoscale size. And when the light falls on that, or if you look them from the different angles, you will get a different color. Like how the butterflies have the various types of the colors. Chameleon, usually we say chameleon changes its color as per its requirement, but it is not like that. It takes the color when the light is fall around on the chameleon. And it has the nanostructure surface. Because of that nanostructure surface, when we look at from it a different angle, we will get we will see it in a different color. How the peacocks, if you see the peacock also, then you will see the different colors, blue, dark blue, green color. And because of this structure present at the nanoscale level, it reflect the color in the reflect the light in different colors. So that is because of the iridescence phenomena, which is mostly related to the nano. So we can say that nanotechnology already exists in the nature, and we are following the nature. If you see, our ancestors also were known to this technology, nanostructures. If you see, first the term nanometer came existence in the 1910. But before that also, around in the century 1200 to 17, there were some steel swords were designed, and they had the different colors. If you look at them at the different angles, you will see the different color. Because this different color, it is not because of the pigment, it is because of the light that is interacting with those nanomaterial. So this is because of the nanostructures. Some potteries were also in the 800 to 1600 century. When they were formed, they were also reflecting the different colors. Some stained glass also. So we can say the nanostructure or nanoscience or nanotechnology, it is not new, but we came to know it about the in the after the 1910. So now we will see the various properties of the nanoparticle and nanomaterial. Why nano is so interesting? Now, if you look at the various research papers and various research going on in the physics, chemistry, or biology, that is mostly related to the nano. Apart from in the basic science, they are doing some research, but all the applied science is roaming around the nano. So why nano is so interesting? It is because the nanoparticles have the high surface to volume ratio. If you take the thing into the micrometer or millimeter size, and if you take the same thing in the nanostructure form or in the form of the nanomaterial, these particles have the totally different properties, and those different properties are because of their height to surface to volume ratio. As we know, as the that the sugar cubes, instead of the sugar cubes, if we take the say, sugar general sugar powder, that dissolves faster. Why it dissolves faster? Because it has the high surface area. And we see in the nanoparticles, because of their small size, they have the high surface to volume ratio. Second thing is that nanoparticles react differently. If you take the catalyst in the form of the nanoparticle, they react differently because their surface has the very high reactivity. Why it has high surface reactivity? Because of the presence of the various atoms on the surface. We will be discussing it later how it will be reacting. Nanoparticles react differently. They also act differently. They have the different behavior with the size. They have the different properties. They interact with the light differently. So when we irradiate the nanoparticle with the light, they behave the differently. You can look into the picture here. Those all are the gold nanoparticles. But if you see, you can see them in the different colors. Why? Because the size is as the size is increasing, it is moving toward the violet color. But if you decrease the size, they will be moving towards the more reddish color. So you can see that the color of these gold nanoparticles is quite different as the size is changes. Next is they are also on the scale of the biological structure. We have recently in the last two slides we saw that the nanoparticles, the size which is most important part of the properties of the nanoparticle is its size, that is 1 to 100 nanometer. And in the 1 to 100 nanometers there are lot of biological molecules like antibodies, proteins. They have the similar kind of the scale of the size, so they can rapidly interact with those biological molecules. Now coming to the how the properties of these particles are changing, how the particle, if we convert a bulk material into the nanoparticle, how their properties are changing. First, there may be change in the shape, and in the if you see it is the bulk material. When we reduce the size of this material into the nanoparticles, we can get the cubes, rectangles, hexagonals, rod shape, sphere shape, needle shape. Various kinds of the shape can be formed. Second is the optical property. Optical property of the bulk material and the nanoparticles are totally different. Just now we saw the example of the gold nanoparticles. Similarly, here is the example of the cadmium selenium quantum dots. You see the nanoparticle having the 2.3 nanometer in size. They have the quite different photoluminescence as compared to the nanoparticles or the quantum dot with the 5.5 nanometer. Very small change in the size, but their optical properties are quite different. So here only the changes of the few nanometers, but if we convert the bulk to nanometer, then there will be a significant change in the size. So the nanoparticles have the different optical properties as compared to the bulk material. Similarly, the electronic properties of the material will also change if you convert it from the bulk to nanoparticle. Like if there is the, as you will reduce its size, there will be the increase in the band gap. I think most of the you science student know what is the band gap. Band gap is the different energy difference between the valence band and the conduction band. So as we decrease the size of the material, there will be the increase in the band gap, and because of that band gap, their electronic properties are changed. Similarly, if we convert the bulk material into the nanoparticle, then the magnetic property of the particle may also change, like iron oxide. If we convert the bulk iron oxide into the iron oxide nanoparticle, they will be converted into the super paramagnetic nanoparticle, which have the which are generally being used in the MRI imaging, magnetic resonance imaging. You might have heard about that technique, which is the most commonly used, or you can say most reliable technique for the diagnosis at present. Next is the mechanical property. Mechanical property of the bulk to the nanomaterial is also significantly changes. Their tensile strength, Young modulus, those things are completely changed from when we convert a material from the bulk to the nanoparticle. So we can say that the nanoparticle, as we convert a material from the bulk to its nanoform, its shape, optical properties, electronic properties, magnetic properties, and mechanical properties are completely changed. Now, how the most important thing, as I told, why we are more looking for the nanoparticle and why they are becoming more important, because of this high surface to volume ratio, and there's particular that 1 to 100 nanometer in size. If you see, here is an example showing that if a particle having a diameter of the 100 nanometer, then how the volume and surface area is changed. So if we calculate for the a spherical particle having a diameter of about 100 nanometer, its volume will be 5.24 multiplied by 10 raised to power minus 22 meter cube. I think you all are aware about this volume for a sphere, that is 4 by 3 pi r cube. So if we put that 100 nanometer here, and then we will get the volume. That volume is about 5.24 10 raised to power minus 22. And if we calculate the surface area for this particle having a diameter 100 nanometer, because it is a sphere, it will be 4 pi r square, and we will get a surface area around 3.41 into 10 raised to power 14. So now if you see, for all the particles, suppose a particle having the size of 1 nanometer, 10 nanometer, here is the calculation which is showing that if we decrease the particle size, there will be the surface area will increase. Here is the surface area 3.14 e to power plus 12. So, and it is changing, it is increasing significantly. On the other side, volume is also increased, but if you look at the ratio, surface area by volume ratio, for a particle which has the 1 micron meter, it is 0.006, but the surface area to volume ratio for a particle having the 1 nanometer size, it is 6. So you can see that how the significantly the surface to volume ratio is changing. And as the surface to volume ratio increases, the percentage of the atoms at the surface and the surface for becomes more important. As the surface to volume ratio is increasing, there will be the more number of the atoms will be present on the surface, and because of that, the nanoparticles have the high surface area, surface reactivity, because there are the more number of the atoms we are present on the surface, and because of that they will be having the more surface reactivity. Now, what is the nanotechnology? Now, if we what we have learned till now from, and how we can conclude this, what is the nanotechnology? So, nanotechnology is the control of the matter on the atomic level. because in atoms, if you see, the size of the atoms is around in the angstrom. So if we take a particle having the 1 or 2 nanometer, it is nothing but a group of the atom. So nanotechnology, we can say that we can control the material at its atomic level. Second is, nanotechnology has the ability to build using atoms as the building block. We can collect several atoms and then we can combine it to form a material. And next, nanotechnology is the manufacture of the novel materials with novel properties. Once you decrease the size, once you are decreasing the size from the millimeter to the micrometer and then to the nano level, the properties are totally different. You will get a material, the material which has the size in the millimeter size, and a material, the same material in the nano form, it will be having the totally different properties, and because of that we say those are the novel materials. Next is the what are the various types of the nanostructures that we can generate. So if you see the types of the material, we can classify these materials, nanostructure or the nanomaterials, on the basis of the dimensions. Like dimensions, they may be the 0D, like the quantum dots or fluorine gold nanoparticles. They are the 0D materials. And this dimension 0D, 1D, 2D or 3D, that we generally depends on how many in dimensions those materials have in the nano scale. For example, the nanotube, you can see it is a long tube. This size in this dimension, the size will not be in the nano scale, it will be in the micrometer or it may be in the millimeter also. But the other size, on the other side, it might be having the nanoscale size range. So in the one dimension, whatever generally the nanotubes, wires, rods, they have the one dimension. We call them the one dimension tubes because they have the nanoscale size in one dimension. If the 2D materials, generally we call the sheets. These generally the graphene sheet, carbon coated nanoplates, the thin films, those are known as the 2D materials. And the 3D materials comes like the spheres of the liposomes, polymer based nanoparticles, dendrimer, because they have the all the 3D structure. Their full 3D structure is in the nanoscale size range. So we can classify the nanomaterials on the basis of the how many dimensions are at the nanoscale: 0D, 1D, 2D, and 3D. Next is the classification on the basis of the materials. We can also classify the nanomaterials or the nanostructure on the basis of the materials by which they has been made up of. For example, the first class may be the polymer-based nanostructures. So we use the polymer for making the nanostructures. Like PLGA polymer is there, polylysine polymer is there, various types of the PLLA, PMMA, various types of the polymers are available. Some synthetic polymers are there, some natural polymers are also there, like dextran. Those are the natural polymer pectins which are also present in the plants, and we can make their nanostructure. So we can convert those polymeric material, bulk polymeric material, into the polymeric micelles. We can convert them into the polymeric nanosphere, and we can also convert into them the nanocapsules. So micelles, you might be knowing the micelles is the amphiphilic molecule. So if a polymer having both the if a polymer has a amphiphilic property, having the both hydrophobic hydrophilic chain, like the poloxamer, they can be easily converted into the micellar form. Otherwise we can convert them into the nanosphere or the nanocapsule form. So we can synthesize the nanostructures using the polymers. Second is the lipid-based nanostructure. We can use the lipids. Lipids and polymers are generally used when we use the nanomaterial for the biological applications, because they are highly biocompatible and non-toxic material. Because if you want to use the nanoparticles for the biological applications, then your nanostructure should be biocompatible. biocompatible. This should not be the toxic. So as our body is made up of the mainly phospholipids, so we can use these various types of the lipids which are already present in the our body. Similar kind of the lipids we can use for making those nanoparticles. Like liposome is one type of the lipid based nanostructure. We can make the emulsion formulation using the lipids, and we can also form the solid lipid nanoparticle using the physiological lipids like stearic acid, glycerol monostearate, tristearine, palmitic acid. A range of the materials are available, lipid materials are available, which can be converted into the nanoparticles. The advantage of polymer and lipid based nanoparticle is there mostly the biocompatibility to the biological system. Next is the protein-based nanostructure. As our body is also made up of various proteins, and various types of the proteins are present already in our body. So we can use the protein for making the nanoparticles for the biological application. So here is the protein which is present in the solution form. So its size will be in the angstrom level, and or sometime it may be 1 or 2 nanometer. But we can aggregate these you can say the molecules of the proteins into a nanoparticle system. You by changing the pH, temperature, or by cross-linking with the other molecules, we can convert these proteins into the protein nanoparticles. So these protein based nanoparticles will be totally biocompatible, and they will not be harmful to the body. So we can use these protein based nanoparticles for the biological applications. Next is the metal oxide nanoparticles. We can convert some metal or metal oxide, like metal, you can say platinum, gold, silver, and zinc. These metals can be converted into the nanoparticles. Some oxides like iron oxide, manganese oxide, those materials can also be converted into the nanostructures. Here is the showing the iron oxide nanoparticle with 5 to 30 nanometer in size. Various size of the iron oxide nanoparticles and gold nanoparticles are there. So these metal oxide nanoparticles, they are generally used for the diagnostic purpose because they give the good contrast, and because of that contrast, we can image the biological tissues like MRI imaging, CT scan. In MRI, mostly the iron oxide nanoparticles are being used. So we can manganese oxide nanoparticles are also the good contrast agent. So we can use metal oxide nanoparticle for the diagnostic applications. Next is the dendrimers. Now, if you see, there are two terms. One is known as the nanoparticle, another is the nanomaterial. Dendrimers are, you can say it is a type of the nanomaterial. Dendrimer is not like that the material which we have to convert into the particle. It is a synthetic molecule. When we start to synthesize this dendrimer, it will automatically form into the nanoscale size. So what is a dendritic structure? Dendrimer is a Greek word, and which is generally means the branched structure. So if a molecule having a branched structure, we can say it is a dendrimer, and it should have a repeated unit so that we can convert this material into a nanostructure. So this dendrimer has a central core. Then we synthesize these different branches, and there is another some free functional group will be available on its surface for the different applications. So you can say dendrimer is a nanomaterial which is present in the nanoscale size range. Next is the carbon-based nanostructure. In the carbon-based nanostructure, we can synthesize various carbon structure using the carbon as the starting material, like graphene oxide sheets, multi-wall carbon nanotube. We can convert most of them are synthesized from the graphite. Graphite is a bulk form of the carbon, and when we process the graphite into the with the different types of the technique, we can convert it into the carbon nanomaterial like single wall carbon nanotube, double wall carbon nanotube, fullerenes, graphene oxide sheets. So these are some examples of the carbon based nanostructures. Now, how we can synthesize these various nanostructures? So basically, there are the two types of the approaches. One is known as the top-down approach, another is the bottom-up approach. In the top-down approach, what we do, we take the bulk material and we transform into the small particles like grinding, cutting. These are the general techniques. Mostly, we can use a ball mill to grind the material and to convert the micrometer size particle into the nano particles. So this is known as the top down approach. We are starting from a higher size material, bulk material, and then we are converting it into the nanoparticles. Another is the bottom up approach. In the bottom up approach, we start from the molecular level at the which has the size around the angstrom, and then we condense it, or we transform it into the formation of the clusters, and then those several clusters combine together to form the nanoparticles. Simply, you can see, you can understand from here that in the top down we take a bulk material which is converted into the powder form, grinded powder form, and further we can, if we reduce the size, it is converted into the nanoparticles. But in the bottom up approach, we start from the atoms, and these atoms clump together to form the clusters, and clusters join together to form the nanoparticle. So here we are reducing the size in top down approach, and in the bottom up approach we increase the size of the atoms, or the clusters to form them the nanoparticle. The other way of classifying the nanomaterial synthesis is based on the synthesis process, like whether we are preparing in the laboratory. Those are the approaches, and know how we will use these techniques to form the nanoparticle. One is the form is the chemical synthesis. We can synthesize the materials using the chemical reactions, and using these chemical synthesis we can convert by the nanoparticle into the nanomaterial, sorry, we can convert the material, or the material present at the molecular level. We can put some chemical reaction and can synthesize the nanoparticle. Another is the laser ablation technique, that is a physical method where we dissolve the metal into a solvent, and then using the radiations we convert them into the nanoparticles. And third one is the biological synthesis. In the biological synthesis we use the some microorganism or the plant product to synthesize the nanoparticle. For example, generally the metal based nanoparticles or metal oxide nanoparticle can be easily synthesized by the biological approach, because the metals have some charge on its surface. We need to reduce its charge. How we will convert the metal into the metal nanoparticle? The simple phenomena is that we need to reduce the surface charge. Suppose gold has the +4 valency. So if we can convert the Au +4 into the Au 0 valent, then it will form the particle. So we have to use some reducing agent which can convert these ions into the particle. So generally you see, almost every plant has some reducing agent. Those are known as the antioxidant, and using those antioxidant we can reduce those the charge of metal ions to convert them into the nanoparticle. So basically nanoparticle synthesis can be classified into the three methods: chemical methods, where we use the, for in the chemical method we use the chemical like a gold nanoparticle. If you want to synthesize, convert the gold Au +4 into the Au 0, you will be using the reducing agent like sodium borohydride. So sodium borohydride convert that Au +4 into the Au 0, and it will form the particle. In the biological approach, we use the reducing agent taken from the plant source, or the like fungi. Some microorganism also give some reducing agent. So those can be used as the uh, reducing agent to form the nanoparticles. Next is the synthesis of the dendrimer. How can we synthesize? As I told that the dendrimer are the 3D architecture having a central core and then different branching units and finally the surface group on its surface. So dendrimer is a nanomaterial. It is not a particle. So we start from the core and then we attach one monomer unit on its surface, and then we will activate the surface and again attach that molecule. So by this repeated addition of these monomer units on the to the central core, it will form a dendritic structure, branch structure, and this branch structure will take a globular shape as the size is increased. And this size, for example, this G4 palmam dendrimer, if we take the example of polyamidoamine dendrimer, that G4 palmam dendrimer that having the size about 5 nanometer. So this is how the dendrimer can be synthesized. Next is the characterization of the various nanostructure. Now, once we have formed the nanoparticles, how we will know that it is a nanoparticle? So we have to characterize what is its electrical property, what is optical property. So we have the different characterization techniques by which we can determine the various properties of the nanostructures. Like the we will be seeing the various techniques for the characterization of size and surface, how we will determine the size and surface morphology, whether it is a needle shape or the hexagonal shape or tetragonal, which type of the shape is present, what is this, how to determine the surface area, specific surface area. We will be determining the surface charge, electrophoretic mobility, surface hydrophobicity, density, molecular weight determination, their chemical characterization and biological characterization. We will be saying that what are the various techniques available for determining these parameters of the nanostructures. So first thing is the size and the surface morphology. So size is most important. Suppose if you have synthesized some nanoparticle, how will you come to know that it is a nanoparticle? Or if it is a nanoparticle, what is its exact size, whether 5 nanometer or 50 nanometer or 500 nanometer? So we have an instrument or the technique which is known as the dynamic light scattering. So it is based on the Brownian motion of the nanoparticle, because the particles have the size less than 5 micron, nanoparticles have size less than 1 micrometer. So they show the Brownian motion, and because of this Brownian motion, we can determine their size. So here the instrument which is known to determine the particle size is known as the Zeta sizer, made by the Malvern Scientific Instrument UK. They have designed this equipment. This is the Zeta sizer nano ZS, which is generally used to determine the hydrodynamic diameter of the particle. Here we keep this sample. Once this lid will open, there will be a cuvette, and we can put our sample into that cuvette, and we will get the particle size through some calculation. So using this hydrodynamic diameter and this DLS technique, we can also determine the polydispersity index. What is the polydispersity index? The polydispersity index gives us an idea about the dispersibility as well as the size of each particle. If the particle, if the our sample having the different size of the particle, like 5 nanometer, 10 nanometer, 30, 200, 500, then it will be a polydispersed system. If all the particles having the same size, then we call it a monodispersed system. So usually we will like the monodispersed system, all the particles should be of the same size. So we can determine this polydispersity index using the DLS. Next is the nanosite. In the nanosite, this is the other instrument which is generally used for determination of the particle size, and generally determine particle size between the 100 to 200 nanometer. So, but it also determines the aggregation stage. If there is any aggregation in the particle, it will easily tell us about that the particles are aggregated, and it counts. It is basically on the it depends on the counting of the particles. So it what it will do, it will measure the size of each and every particle. DLS was dependent on the scattering of the light. As we will light irradiate on it, it will scatter the light. But this is based on the counting of the particles, and there it will count the each particles for their size. Next is the scanning electron microscope. Scanning electron microscope is this is the instrument. You can see in the picture how it look like, and the SEM is generally used for the determination of the morphology. Size we can determine by the nanosite also, DLS also, and SEM also. Here it is the picture which is showing the gold nanoparticles structure of gold nanoparticles. So we can determine the size of these particles, but more important is that we can also see the particles, so we can determine the shape of the particle. Here the nanoparticles mostly look like spherical in shape, and here is the example of the nanorods. So whether the particles, what is the morphology of the particle, whether they are sphere or they are the nanorod or they are cubic in shape, we can uh, uh, characterize these particles for their morphology using the SEM analysis. And other important thing is that what element is present. If we are,
अरे फार्मर के बच्चे इतना तो
so we can determine the size also. So next is the transmission electron microscope. In the transmission electron microscope,
I request to do not write anything on slides, please. Prachi, why you are behaving weirdly?
okay, next is the transmission electron microscope. SEM, scanning electron microscope. In transmission electron microscope also we can determine the size of the particle as well as the morphology of the particles. So only the difference is the resolution. It is the high resolution instrument, and it is based on the transmission of the light through the nanoparticles. So because of transmission of light through the nanoparticle, we can determine the internal structure and the arrangement of the particles. Generally, we need to put the nanoparticles solution on a grid. We dry it overnight, air drying, and after overnight air drying, we put into the instrument, and we can observe the size of the particle morphology using software. Next is the atomic force microscopy. This is the one of the advanced technique which is generally used for determining the particle size in 3D. Till now, SEM and the TM, we can get only the 2D structure of the material, but using the atomic force microscopy, we can determine the size of the particles in the 3D structure. Next is the X-ray diffraction technique. In the X-ray diffraction technique is based on the diffraction of the X-ray light. Once in the XRD instrument, we irradiate the sample with the X-ray, and there will be the diffraction of the light. And because of that diffraction of light, we will get an spectra for that. Here is the spectra for the gold nanoparticles. So there at the 2 theta angle, there will be we will be getting the peak around at 38 degree, 44 degree, 64 and 77. So these are the standard peak for the gold nanoparticles. So using this diffraction technique, we can determine the physical state also. If we get the sharp peaks in the XRD spectra, we will know we conclude that the particles are crystalline in nature. So whether the particles are crystalline or the amorphous, we can easily determine using the XRD technique. If we are getting the sharp peaks, that will indicate the crystalline nature of the material. Otherwise, if the material is the amorphous in nature, we will not get the sharp peak. We will get a hump like spectra here. So using this technique, we can also determine the unknown material because every material has a specific signature. So you can say the XRD work like a fingerprint for the materials. For the any material, if we do not know, we can run the spectra, and there are already available spectra. If we for most of the materials, XRD spectra for most of the materials are already available. So we can determine which type of that particular material is that. Next is the differential scanning calorimetry. You might be knowing this technique. This is generally used for the determination of the melting points and the various, the property related to the energy of the material. So DSC similarly for the simple material that we run the DSC spectra. Similarly, we can do the DSC spectra for the nanoparticles also. So it will give us the information about whether the polymorphism, how much crystallinity is present, purity we can determine the samples purity, we can determine what is the decomposition behavior at which temperature then the nanoparticle will degrade and what are their melting or the crystallization behavior. So we can thermal characterization we can do using the differential scanning calorimetry. Next is the important instrument that is known as the BIIT instrument for the surface area measurement. It is important, as I told in the starting, that why the nanoparticles are so interesting, only because of their high surface to volume ratio. So to measure the surface area of the nanoparticles, we can use this BIIT instrument. It is based on the BIIT theory of the gas adsorption of the nitrogen, how much of the gas is being absorbed on the surface of the particles. So it measures the total surface area, specific surface area of the nanoparticle, and it will also give the information about the pore size distribution, porosity, how much pores are available in the nanoparticle. That information we will get from the BIIT instrument. Next is the surface charge and electrophoretic mobility determinant. Surface charge is very important for the stability of the nanoparticle. If a particle has the more charge, whether it is a cation or the anion in nature, if it has the more surface charge, the nanoparticle will remain stable into the any media. But if there is the particles having the neutral charge near to the neutral, then it will be difficult for them to remain stable because the particle interaction will be more. So if the particles have the high zeta potential or the surface charge value, because of the high charge, they will repel to each other and the particles will remain stable. This is mostly important for the biological system because once we will suppose if we are using the nanoparticle for the delivering of the drug into the body, and as soon as we inject the nanoparticle, our biological system has the various protein, various biomolecules with the different kind of the charges. So those biological molecules get adsorbed on the surface, or they may interact with those our nanoparticle, and they may agglomerate into the body. So the nanoparticle will not reach to the target site in the biological system. So surface charge is very important, and it can be measured by that DLS instrument, which we saw in the very first technique, dynamic light scattering technique. That also gives the information about the surface charge of the nanoparticle, laser Doppler, anemometry, and velocimetry. Those are generally used for the determination of the electrophoretic mobility. Electrophoretic mobility means velocity of the nanoparticle under the electric current, the electric field, how it behaves. That is known as the electrophoretic mobility. So surface charge and electrophoretic mobility are important for the stability of the nanoparticle. Next is surface hydrophobicity. Surface hydrophobicity is another important parameter because this kind of the properties are needed for the materials which we utilize for the making this super hydrophobic surface. Like how a stent is put up into the heart patient. So we do not want that the stent which has kept into the arteries of the heart patient so we should absorb any kind of the proteins on its surface or it should interact with the biological system. Otherwise, those biological material will block that stent. So in that case, surface hydrophobicity becomes more important. And we want the super hydrophobic material which can be designed using the nanotechnology. So surface hydrophobicity can be measured by this hydrophobic interaction chromatography is there, biphasic partitioning process is there, adsorption of probes and contact angle measurement. By these techniques, we can determine the surface hydrophobicity of the nanoparticle. Next part is the applications of the nanostructures. What are the various applications of the nanostructure? So if you look the nanoscience with the is a you can say the nanoscience is an interdisciplinary science which combines all three basic science, whether it is biology or the physics or the chemistry or the material science also. It is the completely interacting with all these three science at various levels. So if you see into the physics, the optical property, laser property, plasma properties of the materials, those are responsible for the various designing of the materials for the various applications like semiconductor engineering, XRD, SEM, AF. So the in the physics, if you want to use a material for the semiconductor purpose, we can design those materials using the nanoscience and nanotechnology because the semiconductor at the nanoscale will behave differently from the materials having the size of the millimeter or the micrometer size. Similarly, in the chemistry, we use the metal nanoparticle, polymeric nanoparticle, carbon nanomaterial for the various purposes. In the electrochemistry, we are using catalyst. We can suppose generally, if you see the chemical reaction, most of the chemical reactions need some catalyst. But if you use the catalyst having a nanoscale size and that we call the nanocatalyst, then the rate of the reaction is being enhanced significantly. So the nanoparticles are being rapidly being used into the catalysis science. Sensing technology in the various sensors chips, those are yeah, whatever this they are being designed like for the glucose detection or the various types of the sensing of the gaseous materials or the another important biological and chemical material, they can be sensed using the nanotechnology at a faster rate. Similarly, in the material science, we can synthesize the various materials for the different purpose. We will see the what are the various generally commonly used material science in the next few slides. And in the biological science, nanoscience is extremely being applied. The lipid-based nanoparticle, as I told, the lipid, protein, and polymer-based nanoparticles, because they are highly biocompatible, they are rapidly being used in the biological science, mostly in the healthcare sector, in the microbiology, designing of the various nanomedicines. They are being used in the. They are used. Nanomaterials are used for the delivering of the various genetic materials like the CRISPR or the siRNA, miRNA, various types of the genetic materials are being delivered using the nanotechnology. So not only to these three science, in the agriculture also the nanoparticles are nanoscience is being applied for the delivery of the pesticides. So we can see the nanoscience is an interdisciplinary science, and now it is playing very important role in physics, chemistry, biology, agriculture, and the energy sector. Now I will specifically discuss this application of the nanoparticles in the cancer treatment. In the cancer treatment, the nanotechnology has shown the significant advancement. Why the cancer? Whenever you talk about the cancer, there is an you will always get a question that why are we unable to treat the cancer? Do we not have the drugs for the treatment of the cancer? Answer is no. There is the flood of the anti-cancer agent available in the market. Then question becomes more irritating. Then what is the issue? Why are we unable to treat a cancer patient effectively? The answer is the toxicity and the hydrophobicity, two important drawbacks of the available anti-cancer drug. If you look at the anti-cancer drug like paclitaxel, docetaxel, they are highly effective. When we take the paracetamol, the dose of the paracetamol is 500 milligram. But if you look at the docetaxel and paclitaxel, they are effective in the nanogram concentration also. But the issue is the how we will treat the cancer patient. Because the cancer cells are generated from the normal cells, and these cells rapidly become the immortal. So there is only one option that we need to do is the killing of that cell. So if you inject the cancer anti-cancer drug into the biological system, they will kill the cancer cell. They are highly effective at the even your paclitaxel is effective in the picogram concentration. But the issue is it will kill the healthy cells also. So this is known as the non-specificity. Cancer, whatever the anti-cancer drug we have till now in the market, they can't differentiate between the healthy cell and normal cell. Whatever the cell will come into the contact with the anti-cancer drug, it will the drug will kill that cell. So we need to design the drug molecules which can only go into the cancer tissue and can kill the cancer cell. So nanotechnology is helping us in this aspect particularly very much. What we can do, we can take that anti-cancer, highly effective anti-cancer drug and can load into a biologically compatible system like solid lipid nanoparticle or micelles, and then we can attach a targeting ligand on its surface, and that targeting ligand will particularly bind to that cancer cell only. So what we can do using the nanotechnology, we can design a nanomedicine which has a cell made up of the biocompatible material. We can encapsulate drug molecule into that lipid carrier or the protein carrier, and then attach a targeting ligand on its surface. This targeting ligand will help to recognize that cancer cell. Okay, so we can see this targeting ligand will help like a biomarker, and it will recognize the cancer cell. So using this technology, nanotechnology, we can design the better nanomedicine for the treatment of the cancer. Another thing that is we need to monitor the progress of the disease, how the disease is progressing, how the cancer is spreading fast. So for monitoring that, we need the contrast agent. And as I told that the metal oxide nanoparticles, or they are giving the very good images during the imaging in the MRI or the CT scan. So metal oxide nanoparticles, they can be used as the imaging probe or the diagnostic tool in the cancer. So in the cancer, we can say the nanosystems, polymer-based nanoparticle or lipid-based nanoparticle or your protein-based nanoparticle, they can help us into designing of the nanomedicine that can be used in the therapy for delivering of the chemotherapeutic agent or proteins or the peptides. On the other hand, metal oxide nanoparticle can be used in the imaging. Now, where we can see the nanotechnology? Have you seen the nanotechnology around in your day-to-day life? Here are the few examples where we can see the nanotechnology in our routine life. This is a tennis ball, and now some tennis ball has been designed on the nanotechnology base. Here, these are the specific tennis ball which have a nano composite gas barrier. Generally, what happens when we play with the ordinary tennis ball, the air inside present inside the tennis ball will come out with the as we play with the ball. But if we put in the newly designed tennis balls, they will come. They contain the nano composite gas barrier, and because of that, this gas barrier, yeah, the gaseous molecule will not come out from the ball, and its life of that ball will be more as compared to the ordinary ball. Here is another example of the tennis racket. Now the rackets are being designed using the graphene oxide, because graphene oxide is a carbon material and it has the high mechanical strength. Two important things is there. One, that the carbon nanotubes are have the very high mechanical strength, as well as they are light in weight. So to design the lighter racket, badminton racket, they are now being prepared using the carbon nanotubes. This ad you might have seen, the Lifebuoy ad every day we can see in the advertisement on the TV. So here you will be seeing this in this ad. They will always talk about the silver ions, because they say it is showing the 100% germ protection and the new silver. And that new silver is present in the Lifebuoy, so in the nano form. Next is this the another kind of the thing, that is the self-cleaning cloths. There are the some companies which are making the cloths which are the self-cleanable. Why this is based on the lotus effect. As I told in the very first slide, that the how the lotus always lie in a dirty place, but its leaves will always remain clean. Why they are clean? Because of the super hydrophobicity. So companies have designed these self-cleaning cloths. So if we wear and if we go out, and if we even it is raining, we do not bother about that, because immediately that raindrops will be they will not stick to the cloths, and it is easily cleanable. And even if something fall on the dirt, that will also not stick to the these cloths, because of the super hydrophobic nature of these cloths. So they are self, we can say those are the self cleaning cloths. Nanotechnology is very common in the cosmetics. Zinc oxide, titanium oxide, polymeric nanoparticles, those are mostly being already, gold nanoparticles, they are already being used in the nanoparticle in last 2 decades. Zinc oxide and titanium oxide, they are used in the sun screens. Hyperbranch polymers are being used in the mascaras. Solid lipid nanoparticles are used in the perfumes and acne treatment. In anti-aging creams, they contain the zinc oxide nanoparticles. Some hydrogels are present in the face mask. And similarly, in the various hair products, they contain the anti-UV products, so that those will not affect the hair. So nanotechnology is all around us. And yes, another next example is the quantum LED. You might have heard about this Samsung QLED. It was launched about 5-6 years before, quantum LED, and they are made up of the quantum dots of the size around 2 to 5 nanometer. Recently, LG also launched the nano cell. Those are also made up of the quantum dots. So this is the example of the application of nanoparticles in the physics. And yes, next there are some socks also, nano socks. Those are known as these socks contains the nano silver. So because of the antibacterial effect of the silver nanoparticles, that which we have seen in the Lifebuoy ad also. So because of that, and another thing is that they will be containing the anti-radiation nanoparticles also, so they will protect them from the radiations also. And some germanium stones are also present in these socks, so that will give like the acupressure kind of the acupressure effect. So what we can conclude from here is that the nanoscience and nanotechnology is a new and rapidly growing field with a very wide range of the applications. It is being used in all the sectors, whether it is energy, healthcare, electronics. In each sector, agriculture, they are being used. Various technologies till now these researchers have developed to synthesize and characterize the different nanostructures. We have seen that the AF starting from the DLS, nanosize, then SEM, TM, AFM, XRD, DSC, electrophoretic mobility analyzer, then your BT surface area analyzer. A range of the technologies has been developed to characterize these nanostructures. And we have a wide range of materials also. We can produce the nanostructure from the various materials like polymer based material we can synthesize. We can use the lipids for the synthesis of the nanoparticle. Carbon based nanoparticle materials can be used for the synthesis of nanoparticle. You can use proteins for the synthesis of nanoparticle. And the other basic using the different small molecules, you can synthesize the dendrimer like structure also for the synthesizing various nanostructures. And nanotechnology has finally the nanotechnology has entered in our everyday life, and it will change it significantly. So thank you. Stay home, stay safe. And now it's time for the interaction. If you have any queries, please let me know.
Yes, Priyanka, if you have any question.
Yes, ma'am. Good evening, sir.
Yes, good evening.
Sir, actually, about nanoparticles, sir, we know that it is included in our everyday life. But the thing is, I want to know about the toxicity levels of nanoparticles, sir.
Yeah, as I told that the nanoparticles, specifically the metal oxide nanoparticles are quite toxic. So when if we want to use those nanoparticles for the biological applications, suppose if you want to use the metal oxide nanoparticle for the making the fuel cell or the semiconductor material like TV, microphone, or the mobile, then it's okay. But if you want to use them for the biological application, they are quite toxic. So generally, it is recommended that the nanoparticles, whatever we want to use for the biological application, we should use specifically the lipid-based nanoparticle or polymer-based nanoparticle, which are the polymers like PLGA. PLGA is made up of the polylactic acid. And if we remember the Krebs cycle, there is the lactic acid and glycolic acid. Both are the part of the Krebs cycle. So if we use the PLG and nanoparticle, that PLG will degrade into the biological system through the Krebs cycle, and the ultimate product will be only the carbon dioxide and the water. So we have to be careful when we are selecting our nanoparticles depending on our application. Now, on the other side, if you see the metal oxide nanoparticle, they are quite stable nanoparticle. They will not degrade in the biological system, so they may be toxic. But if you want to use them for the diagnostic purpose, so like a MRI, how many times we go for the MRI? Generally, up to the 40-45 years of age, nobody goes for the MRI because until unless there is a specific cases. So for that purpose, in the our whole life, we go only for the once or twice for the MRI imaging. Then the nanoparticle are good. Metal nanoparticles are good for diagnostic purpose because they will remain stable in the biological system, and we need not to inject them again and again for monitoring of the disease.
Thank you, sir. Sir, I have one more. Can I ask, sir?
Yeah, yes.
Sir, actually, I want to know the other fields where we can explore nanotechnology very well, other than the fields that we are using it now, like it may be for agriculture, it may be biotechnology or forensic, whatever. The other fields which is not explored yet.
No, most of the structure, because the nanoparticle has nanoscience is rapidly growing in last 20 to 30 years. And because of their so enormous and excellent properties, everybody wants to apply that technology into their own system. So till now, I do not look that any field is remaining that it is not being explored. In the rocket science also, they are using the nanotechnology to design the light materials like graphene oxide, which remains the stable and the lightweight also. In the agriculture, healthcare, energy, cosmetics, pharmaceuticals, every field is using the nanotechnology. So the only thing that remains is that what we can do with nanoscience, the new into the in whatever the science we are studying as our basic science, or botany, zoology, or any kind of the biological science or the material science, how we can apply this nanoscience into our research.
Yes, sir. Thank you, sir. And I recently went through some article in journal, sir, which gave me information about using nanoparticles as regeneration medicine, sir.
Yes, nanoparticles are used in the regeneration medicine. Suppose if somebody gets the somebody catch an accident, and if there is some damage of the tissues, by that time what we can do, we need to put that stem cells at there. Usually what happens when we get hurt from something, immediately our body will form the new cells. So similarly, if there is a major accident, and in that case, if the person has lost some part of its body, by that time we need to generate the doctors need to generate those areas very fast. So that to improve that healing process, what they will do, they put that stem cells at that place, so that that area will be healed quickly. So to improve these things, nanomaterials can help, like the porous nanostructure. If we use the porous nanostructure, then we can put those stem cell on that porous structure, and we can provide the feeding material. Whatever the media is required for those stem cells, that we can fill into those porous structure, and we can place into the damaged part, and it will give the strength to that part, and slowly the stem cells will grow at there, and they will form the tissue, and we can use that nanomaterial, porous nanomaterial made up of the polymeric for various kinds of the polymers, which have the mechanical strength. Also they have the biodegradable property. So slowly that nanomaterial will degrade into the body, and the stem cells for which it was working as providing the mechanical strength, that will help to generate the new stem cells.
Thank you, sir. Thank you so much.
Regenerative medicine. Yeah.
Good evening, sir. Sir, I have a
Yes. Yes, please.
sir. While studying
Why? Sorry.
So Saksham this side. Meanwhile, maybe got his mic cut off. So I had a question.
Yes.
So usually when we are using nanoparticles in biological system, they are capped with a conjugate pair of polysaccharide or any. They are capped with
Yes.
polysaccharides.
Okay.
Yeah. So which technique can we use to characterize them, like the capped nanoparticles?
So generally, if we use these nanoparticle for the biological system, they can be kept for the various purpose. So for example, one of the major reason is that to stabilize those nanoparticle into the biological system. Suppose if you want to use the manganese oxide nanoparticle or iron oxide nanoparticle for the diagnosis of particular cancer. So during that purpose, because metal oxide nanoparticles are not stable into the biological system, you can cap with the biocompatible material like polysaccharide or some proteins or the some polymers which are highly biocompatible. So once the coating is done, then we need to characterize them for how much size has been increased, what is the surface charge has, how the surface charge of the particle has been changed, and how much coating is there. So coating percentage that can be determined by this TG analysis, thermogravimetric analysis. The coating percentage we can determine, particle size and shape we can determine the SEM, and only the important thing is because that how much coating has been done on the those nanoparticles. So because the coating percentage will definitely change their behavior in the biological system, like where they have to go, how they will be degrading into the biological system. So that is most important in the coated material to know the coating percentage.
Okay, sir. Thank you, sir.
Sir,
Yes, please.
sir, I want to add to that question. So when you tag polysaccharides or proteins to these particles, wouldn't that induce a immune response in the body when you like put it into the biologic, like the human system?
No, I did not get you. What is your question?
So when you cap these particles with polysaccharides or proteins,
Protein. Okay.
and when we introduce it into the body, wouldn't that induce an immune response?
Immune response, yeah. But if you induce these, if you inject the particle, blank particles, body will take it as a foreign material, and it will behave. All the nanoparticles will behave as a foreign material. So the mononuclear that we MPS system, phagocytic system, immediately may get activated, but it has been observed that if the particle size of the if the size of the particle is less than 200 nanometer, they deceive our macrophage system in in our body. So macrophages can't recognize a particle having a size of the less than 200 nanometer. So it is an important point when we are using the nanoparticles for the biological application. We try to keep the particle size below 200 level, 200 nanometers, so that our macrophages can't recognize, and it will help to it will help us to avoid that immunological responses that may occur in our body.
Sir, it may also depend upon the route of administration also, no?
What?
I mean, sir, when it's at the nanoparticles or the medicines are administered through a oral route, then oral route is more like a tolerative route with respect to immune system. So
It depends.
wouldn't that
It depends
immune
what
response
is our
depend?
final objective, where you want to deliver, whether you want to deliver into the particular organ, like the colon cancer. In the colon cancer, if you want to deliver the drug for the colon cancer targeting, then the nanoparticles which are being introduced through the IV route or the oral route, their behavior will be totally different, and the amount of the nanoparticle reaching to the cancer cell will be different. So we have to be very careful when we are designing the nanomedicine. Obviously, the route of administration will definitely affect. For example, if you want to target the lung cancer, then oral route is better, because and you use the solid lipid nanoparticle, because they are transported through the M cell. They will reach into the lymphatic system first, and they will directly deliver to the lung. So we need not to target those nanoparticles to attaching another targeting ligand. So lipid-based nanoparticles, they have the phenomena or the different phenomena of absorption through the intestinal system. They have the M cells available for their absorption, and through the lymphatic system, they will reach to the lungs. So it depends. Okay, yes, definitely the route of administration affects the biodistribution of the nanoparticles.
Thank you, sir.
Hello, sir.
Yes.
Good evening, sir.
Your
Sir.
voice is very low.
Hello, sir.
May you please increase your volume of your system?
Okay, 1 second. Hello, sir. Now,
Yeah, it's audible now.
okay, sir. Sir, during the synthesis of nanoparticles after milling process used through like sir ball mill. So, sir, sieving process is also required, sir. For
Sieving?
sieving process, sir, like sir size separation from the coarse particles. After that, sir, milling process.
Yes. She is because of the nanoparticle in the ball mill. They have the different. They will be having the different size distribution. There will be the particles from micron size to the nano size. So, when you are using this ball milling for the reduction of the size, you have to optimize the various parameters like what will be the speed of the ball, the ball rotation speed of the of the ball mill, how long you have to rotate it, and the sieving process definitely help us to get the smaller particles separated from the larger particles.
Okay, thank you, sir.
Hello, good evening, sir.
Yes.
Sir, I have a question that how we can determine the loading efficiency and drug loading capacity of any missile.
In the micelles. So in the micelles, if you see, there are the two basic process by which you can determine the drug loading and the drug entrapment efficiency. So one thing is that once we put the drug into the loaded into the micellar system, the excess of the drug will precipitate out. So you can use the centrifuge technique to separate out the unloaded drug that will settle at the bottom, and in the supernatant you will get the micelles. Another technique is the filtration. Using the 0.22 micrometer filter or 0.45 micrometer filter, you can filter your formulation so the unloaded drug will remain on the filter, and in the supernatant you will get your drug loaded nanoparticles. So the two techniques by which we can. So do you want to know what is the drug loading or drug entrapment efficiency as well?
Yes, sir. Actually, the drug loading capacity,
Yes,
how we can determine it.
yes. The thing is that there is the misconception between the people that drug loading and drug entrapment. Both are the different things. Drug entrapment is totally different from the drug loading.
Yes,
Drug
sir.
entrapment means, suppose if you are using the 100 milligram of the polymer or lipid and you are adding the 10 milligram of the drug. So how much of the drug has been entrapped? Suppose your system is able to entrap the 9 milligram of the drug. We added 10 milligram of the drug, and we observed that the 9 milligram of the drug has been entrapped. So their
Yes.
entrapment efficiency will be the 90%. But in case of the drug loading, in the drug loading, we also consider the weight of the polymer that we have taken. So is the drug loading will be the 9 divided by 100 plus 9, because we have taken the 100 milligram of the polymer or lipid also. So in first case, there will be 9 divided by 10 multiplied by 100, 90%. In second case, there will be the 9 divided by 100 plus 9 multiplied by 100, and it will be 0.82%. Sorry,
Okay.
8.2%. So entrapment efficiency, where you consider 90%, loading efficiency is only 8.2%.
Okay. Thank you, sir.
Hello, sir. I am Swapnil from Maharashtra.
Yes, sir.
Sir, my question is that why does the fuse keep blowing during the hot pressing of nano powder?
May you please repeat your question?
Why does the fuse keep blowing during hot pressing of nano powder?
I did not get you.
Sir, I'm pardoning now. Why does the fuse keep blowing during the hot pressing of nano powder?
Can you elaborate little?
Ma'am, I am facing problem. The fuse keep blowing during the hot pressing of nano powder. I didn't reason behind that.
No, what is that? What are you doing and what is happening? Can you just elaborate it?
The fuse. Why does the fuse keep blowing during hot pressing of nano powder? Means, sir, fuse like means electric fuse.
Hmm.
when I spread out the nano powders, they blowing. The fuse is blowing.
Fuse is blowing.
Yes, sir.
Even I didn't understand.
I didn't. Yeah, I am unable to understand what is your question exactly.
Can you reframe it after that? And meanwhile, if somebody has question.
Yes, ma'am.
Sir, there is a question in chat box that why nanoparticles are not used in large-scale agriculture in India.
Yes,
Discuss your views on nano-sized
yes.
manganese calcium cluster in photosynthesis.
Yeah. The thing is, in the agriculture sector, till now the nanoparticles are only at the, you can say, the exploration stage only. They at the research level only. They are not most at the practical use. The reason is that the government or the agriculture sector, that is the chemical and the fertilizer sector, they are more, they have the concern regarding the toxicity of the nanoparticle. What happened during the Green Revolution? We started to use the pesticides at the large scale immediately, and now we are facing the problems like the cancer. Cancer because of this green revolutionary and the revolution and the use of the pesticides in our the agriculture. So so there are some toxicity concerns with the nanoparticles also. If you see the metal oxide nanoparticles, they are already toxic. We know that they are the toxic, and we do not use in the biological system. We use the lipid and polymer based nanoparticle, but our body can biodegrade those nanoparticles, but there is no phenomena that can degrade those lipid or polymer based nanoparticles also in the environment. So because of that, they are not being explored at the for the practical use. Now slowly some industries are starting at the, they have taken the few fields on the rent, and now they are evaluating the toxicity in the real biological real environmental system, and to explore their toxicity, how the environment is being affected. Hello.
So I had another question.
Hmm?
So recently I was reading a review paper on nanoparticles. In that paper, there was a parameter that was like that the time of storage of nanoparticles. So in the process of basically as a parameter to process nanoparticles or to make nanoparticles, it was stated that the time for being that it's stored is a dynamic time. That means it has a dynamic effect. That means some nanoparticles may shrink or some nanoparticles may become larger with the increased time.
Right.
So why does that happen, sir? I mean to say, sir, if we are storing for a longer time period of time, then why are they shrinking or?
Are those nanoparticles for the biological application?
Sir, I'm not sure because I was just reading the parameters.
Okay. So
Yes, sir.
suppose if we take the example of the biological system, the stability is most important parameter, because suppose you are making a nanoparticle of the doxorubicin drug. So there are two types of the stability most important. One is the colloidal stability that we call the colloidal stability, and another is known as the storage stability. So what is the storage stability? Suppose a pharmaceutical industry today, they are manufacturing the doxil, which is already available in the market, doxorubicin's nano formulation. So suppose they are manufacturing it today. How long it will take to reach to the patient? It will take a very long time. They are manufacturing today, then it will be they will distribute it to the wholesaler, then wholesaler to the retailer, and patient should go to the doctor will prescribe the drug, then it will the patient will go to the retailer and he will take that drug molecule or that nanomedicine. So it is a long process. So it is very important that the nanoparticles formulation should remain stable for a longer time. Storage stability should be like at least 2 to 3 years, so that they are not expired very quickly. So in that case, the nanoparticles are supplied in the form of the lyophilized particle. There will be no water in the that form. It will be the solid particle. Lyophilization, you might be knowing that freeze drying technology they use. And they supply those lyophilized powder with the water for injection. So what will the doctor do? Once doctor patient will come to the doctor for that injection, he or she will add that water for injection into those nanoparticle, lyophilized solid nanoparticle, and he or she will shake that particle and will inject to the patient. So two important things. First was the storage stability, and once as he dispersed those nanoparticles into the water for injection, they should remain stable during the injection process as well. So the important property is that the size of the particle should not change, surface charge of the particle should not change, polydispersity or index should not change, and the drug content, the drug quantity that is present in that form, that should not affect. So these are the four basic parameters that are generally used to determine the stability of the nanoparticles. So if they are using in the pharmaceutical application, so they need to check the nanoparticles should not change at the various stages. Like they test those nanoparticles at the 25 degrees centigrade, slightly high temperature, like at around 45 degrees centigrade. As per the ICS guideline, they check those nanoparticles, that whether the nanoparticle are forming aggregate or precipitate or any kind of the deformation, that should not happen into the nanoparticle.
Okay, sir. So it's basically stability
Yes,
that is making them.
stability is very important for specifically for these pharmaceutical products.
Okay, sir. Okay. Thank you very much, sir.
Okay, so further question? So I consider there are no more questions, and if you have any, then you can drop down your question or query in the WhatsApp group, or you can mail me. Thank you so much, sir, for your time, and we are really glad to have you, that you have accepted the invitation just before one day, and it's very nice of hearing you. It was really informative, and I think by seeing the discussion only, we can understand that students get very, student finds it very useful for their current studies. So thank you so much, sir.
Thank you.
Yeah, and we always look for your support for such future